Hormonal control of spermatogenesis follows the general hypothalamic anterior pituitary gonadal axis. So this is the HPG axis. So basically, there's pulsatile release of gonadotropin releasing hormone. Gonadotropin releasing hormone from the hypothalamus then stimulates the anterior pituitary to release the gonadotropins, follicle stimulating hormone and luteinizing hormone. These two hormones are then released from the anterior pituitary and then travel towards the gonads. In this case, we're specifically looking at the hormonal control of spermatogenesis. So they are traveling to the male gonads or the testes. The specific target for follicle stimulating hormone from the anterior pituitary is the Sertoli cells. So although FSH, or the primary role of FSH, is spermatogenesis and the development of the spermatogonia into the mature spermatozoa, the actual spermatogonia do not have receptors for FSH. The receptors for FSH are located on the Sertoli cells. So the target of FSH is therefore the Sertoli cells. And once FSH then binds to the membrane-bound receptor on the Sertoli cells, it initiates second messenger systems to then initiate the production and the synthesis of various paracrine molecules which are required and needed for the development of the mature spermatozoa. So it's needed, all of these paracrines and molecules which are then produced by the binding of FSH are used to send the germ cells or the spermatogonia into the mitosis, so producing more germ cells, and then moving on from mitosis into spermatogenesis, so into the meiotic divisions. In addition to the different paracrines which are produced through the binding of FSH, which help with the process of sperm production and development, FSH, or the binding of FSH, also leads to the production and release of androgen binding protein. Remember, we said androgen binding protein binds to testosterone, making testosterone less lipophilic and thereby keeping testosterone within the tubule lumen of the seminiferous tubules. Another protein that's also produced by the binding of FSH is inhibin. Inhibin then activates the feedback mechanism and then inhibits the production of further FSH. So inhibin serves as the negative feedback signal. And to the other end, the primary target of luteinizing hormone is the Leydig cells. Remember, the Leydig cells are located in the interstitial tissues or interstitial fluid, so not within the seminiferous tubule. And the Leydig cells, as we've mentioned, produce testosterone. So testosterone is essential for the process of spermatogenesis takes place. But the actual actions and effects of testosterone are mediated by means of the Sertoli cells, because the Sertoli cells have testosterone receptors, but the spermatogonia do not have testosterone receptors. So testosterone cannot directly bind to the spermatogonia or the spermatocytes because they lack these receptors. So testosterone's effects are then mediated by means of the Sertoli cells. And testosterone then initiates the negative feedback mechanism to inhibit the luteinizing hormone and then also the production of gonadotropin releasing hormone. After the production process and the development process of sperm, the sperm then leaves via the epididymis, which then becomes the vas deferens and then travels to the urethra. Now this process is called emission. So the pathway traveled from the vas deferens to the urethra. Once it reaches the urethra, sperm is then joined by various secretions from the male accessory glands. Now these secretions come from the seminal vesicles, the prostate, and the bulbourethral glands. So this then forms a sperm fluid mixture which is known as semen. When semen in the urethra is then expelled to the exterior, it is called ejaculation. So there is a difference between ejaculation and emission. Each ejaculate is about 2 to 6 mils, and the main nutrient that sperm uses for energy is fructose. This table just basically summarizes all of the different components of semen. So yes, we know that semen is composed of the sperm and then all of the different secretions, but then this is further subdivided. So sperm we know comes from the gametes, which is actually produced specifically within the seminiferous tubules, but then also there are other components including mucus, water, various buffers, nutrients which all nourish the sperm, which basically come from the seminal vesicles, the prostate, the epididymis. Then there are also various enzymes which act in clotting the semen in the vagina and then also liquefying the clot. There's also zinc. Zinc is actually an interesting component of semen. Its role or its exact role in reproduction is not clear, but it has been found that when the concentration of zinc is actually below a certain level, it leads to infertility. And then another component is the prostaglandins. So the seminal vesicles contribute prostaglandins, which actually appear to influence sperm motility and transport. Okay, so we've basically focused on the role of androgens and how essential they are for the primary sex characteristics. So in other words, we know that they're important for the development and differentiation of the internal and the external genitalia. Androgens are important for gametogenesis, etc. So basically, when we talk about primary sex characteristics, we talk about the primary characteristics which distinguish males from females. But androgens also have a number of effects on the body in addition to these primary sex characteristics, because they're also involved in secondary sex characteristics. So in other words, all the other traits which distinguish males from females. These include body shape. So the typical male body shape is an inverted triangle, so broad shoulders with narrow waist and hips. Androgens also are responsible for the typical male traits such as growing of a beard and body hair, as well as muscular development, the lowering of the voice, which is caused by the thickening of the vocal cords, and then androgens also contribute to various behavioral effects such as sex drive or libido.